So good evening, everybody. Welcome to this international webinar at Believers Church Medical College and Hospital. Today, it's a fascinating topic, and it goes like this: finding a shortcut, a hitchhiker's guide for nanoparticles to reach the peripheral nervous system. Well, any drug related to the nervous system. So good evening, everybody. Welcome to this international webinar at Believers Church Medical College and Hospital. Today, it's a fascinating topic, and it goes like this: finding a shortcut, a hitchhiker. Sorry, there was a glitch. Sorry, there was a glitch with the system. We'll resume. So this is your topic for the day: finding a shortcut, a hitchhiker's guide for nanoparticles to reach the peripheral nervous system. So the nervous system per se is fascinating, and any drug that reaches the nervous system is a game changer. So nanoparticles are now in vogue and very much are game changers in various therapeutic strategies. So we have a great speaker from Amrita Institute of Medical Sciences. He's a postdoc from Harvard, and he has chosen his home country to come and work. And it's none other than Assistant Professor at Center for Nanosciences and Molecular Medicine at Amrita Institute of Medical Sciences, Kochi, Dr. Sahadev Shankarappa. After completing his MBBS from Bangalore University, he went on to do his MPH at the prestigious University of North Carolina at Chapel Hill, and went on to complete his PhD in neurosciences from Loyola University, Chicago, and completed his postdoctoral fellowship from none other than MIT and Boston's Children's Hospital, Harvard Medical School. He's a DBT Ramalingaswamy Fellow, and he is championing the cause for nanoparticles for the nervous system. So without much ado, let's hand over the session to Dr. Sahadev Shankarappa, our speaker for the day. We also have our moderator, Dr. Alice David, the head of medical research, epidemiology and biostatistics. Ma'am, it's a great privilege to have you as the moderator. So ma'am, his MSc from the prestigious CMC Vellore, and she completed her PhD in epidemiology from Rutgers University, USA. So she's been extremely competent in research and has been guiding numerous students, faculty, and research scholars across the globe. So it's a great privilege to have her as the moderator. So welcoming both the speaker and moderator, let me hand over the session to our speaker, Dr. Sahadev Shankarappa. Over to you, sir.
Dr. Jacob, thank you very much. That's a very generous and a gracious introduction. Thank you. And it's good to be at the Believers College Hospital here talking to the students and I'm sure faculty. And Dr. Alice, thank you for having me over. And today, before I start, let me share my screen with you. Okay. Is this visible? You can see my screen?
Yes, sir.
Yes,
sir.
yes.
It's visible.
Okay. So As Dr. Jacob mentioned, that the title today that I have chosen is trying to see if we can figure out an alternative route to get to the nervous system. So we work on my laboratory in Amrita. So we work on our interest mainly lies in axonal biology, and we try to figure out how neural inputs from axon determines various different kind of pathologies, if I may. One of the projects that we are looking at is how axons affect the way tumor cells behave. And that's one of the things that we are doing. But a large part also has to do with drug delivery systems and nanoparticles. And our interest lies in trying to see if we can get nanoparticles to neurons. And of course, that has its challenges. And I'll touch upon a few of those challenges in my talk. So from what I understand, I'll deliver my talk, and I've designed it for roughly about half hour or so. And after that, if you have questions, I'm sure there's the system here where you can put in your questions, and I'll try to address them. So before I get into my topic, I just want to acknowledge the institute where I work. This is a very nice aerial picture of Amrita, and now there is a lot more construction going on. This car park here doesn't exist anymore, but we are nestled within the hospital campus. The reason I mention is that now that with newer academic settings coming up and for research going on, it is quite important that clinical people, medical people, and researchers interact. So I'm so happy to see how your college is actually getting these seminars up and calling people from different backgrounds to come in. And that gives a very good exposure for students as well. So I'm glad you guys are doing that. And at Amrita as well, we have many exercises and practices in place where we are trying to increase this interaction between researchers, benchside researchers, scientists, and clinicians and people with medical background. So we are in one of these blocks up here, and is very closely associated with the hospital, and we have a lot of crosstalk with physicians, clinicians, and many of these professionals. Having said that, before I get into my topic, I would like to thank a few people. Without them, these work, what I'm going to show you, wouldn't have happened. These are my lab members, and this was a picture taken pre-COVID era. If we all even remember, we all, I'm sure, fondly remember those times when we could go around and do whatever we wanted to. So this was a picture taken in the pre-COVID era, and hopefully we'll get back to this era soon, maybe this year itself. So I would like to thank my students, Neeraj, Gayatri, Pallavi, and Chinnu, whose Neeraj's work is what I will focus on today, and all the other students who have contributed in their capacity to this work, which I'll discuss. I would like to thank the Institute, Amrita Institute of Medical Science, for the infrastructure and support that they give. And of course, without the funding, the studies wouldn't take place. So I'd like to thank Department of Biotechnology and the Department of Science and Technology for generous funding of our studies here. And there are a few other collaborative grants that we have. So as I've said before, getting nanoparticles specifically are important. I mean, nanoparticles have been around since 1990. And so far, the last I've checked, about 40 nanoparticle formulations have already been put in clinics for various different reasons. Predominantly, it's mainly for cancer that we see a predominant nanoparticle category of drugs that go in there, and also for imaging purposes and things that have been done. But nonetheless, for the nervous system, when it comes down to nanoparticles and other kind of therapeutic cargo, you all know that it's fairly challenging to get particles in. So conventionally, we have your regular type of routes of delivery systems, which includes the oral system, the intravenous, intrathecal, cerebroventricular, and intranasal. Of course, there are a few more, but I've chosen to highlight the more conventional ones. And as you all know, no matter how much we try, one of the challenges that we face when we try to put drugs into the nervous system is what is shown on the right side here, which is an illustration of a blood-brain barrier. So here, the nanoparticles or the therapeutic cargo, once they go into the blood vessels, they find it very hard to come outside. That's primarily because of the extremely tight junction proteins that are found between endothelial cells that makes these particles to find it very difficult to get out of the vasculature and then get into the neural tissue or the neural milieu to cause their effect. And this observation that we see in the central nervous system is no different when we consider the peripheral nervous system as well. So this illustration here as well shows this is a peripheral nerve with all its covering. You might all recall that it has three layers: epineurium, the perineurium, and the endoneurium. So within lies the axons that traverse long distances via the peripheral nerve. The vasculature hugs very closely with the peripheral nerve and even enters the substance of the nerve. Now, within the substance of the nerve, it gives out branches that gets in very close proximity to axons. But however, again, when a therapeutic cargo and nanoparticles tries to exit the vasculature, they're again faced with tight junctions, as you can see in this illustration, to get from the from the vasculature into the peripheral nerve space, so that they can touch upon the axons. So there are a lot of proteins have been characterized. The blood-nerve barrier has been well characterized. We know it's quite difficult to get through. So we need to figure out certain strategies. One of the strategies that we have thought about is, how about the axon? Okay. So as you know, axons are some of the longest cells. The axons, especially in our legs, that goes via the sciatic nerve, it goes about roughly 1 to 1.2 meters. The axon goes that long. So these gives us these routes to the nervous system. Now, could it be possible that such a route could be utilized for delivering therapeutic cargo? So what the idea behind, or the starting idea, what we had thought about, was that if we can develop nanoparticles which can be applied in the proximity of axons, and if these axons can then somehow transport these nanoparticles into the peripheral or into the nervous system. So at this point, we weren't particularly sure the range it's going to take in. I mean, will it go all the way? Will it stop at certain places? We weren't sure. But the idea was that, can we put these particles in and see if then get transported via the axons? That's what we started out with. And to test this, we have utilized the illustration that I've put out here is the dorsal root ganglion system. So as you might all be well aware that the dorsal root ganglion houses the cell bodies of sensory neurons that innervate our periphery. So this was fairly simpler as compared to axons in the central nervous system. We could have tested this thing in the central nervous system as well, but the anatomy, the neuroanatomy within the brain makes it fairly difficult to access axons, and since this was a proof of concept, we had to test it in a fairly accessible system. And so that was primarily one of the reasons why we went ahead with the peripheral nervous system, using axons of the peripheral nervous system and trying to look at the dorsal root ganglia and see if we can deliver axons via these axons from DRG neurons. And the neurons in question are these guys here. You may be familiar with these. These are called as the pseudo-unipolar neurons that are found that are located within the dorsal root ganglia adjacent to the spinal cord. And these gives out axons that goes all the way to the periphery, and the other end goes into the dorsal horn They go into the dorsal horn. And from there, there's a synapse that occurs. And from there, the second order neurons go up via the various tracts, spinothalamic tract and a few other tracts, and they go up into the brain. So we used this particular system. And these neurons, one of the things is that we can isolate the DRG, and we can dissect out the DRG from an experimental model, a rat or a mouse, and these can be cultured, so in plates. So that gives us a lot of opportunity to test our systems first before we try to get into biological systems. And here's a small video I wanted to play. I would draw your attention to these arrowheads where you're seeing these two neurons. And this is a culture that we have done, one of my students has done. And these, within a matter of 3 to 4 days, they sprout axons. So hopefully you can see this. You can see these axons coming out all the way, and they go touch, and they do a whole lot of stuff. It's very fascinating to see them. So there's the axons. So this gives a very nice opportunity to separate out the cell body and the axons, and then try our nanoparticulate systems. Now, the axons themselves, when you look at them under a simple hematoxylin and eosin stain, and you look at them, they have very nice structures, and you can pinpoint if there is something has gone wrong, the axonal degeneration, any kind of lesion, necrosis. It's fairly easy to detect in the peripheral nerve. When we do a transmission electron microscopy image, when you look at it, you see these nice axons in their myelinated ones. There are these unmyelinated axons as well. So the point here is that this system gives us access to see how these axons are doing when we start our treatment, if there's anything going wrong, or if anything can be tweaked. So that's the advantage of this system. And as a reminder again, these have very specific innervations. We have specific dermatomes where we know the migration of nerves that have taken place, and we know which area are innervated by which nerves at what level. I mean, these things are well known. So that also makes it a very interesting concept to see if we can use therapeutic molecules later on for specific areas to be transported to specific regions within the nervous system. Now we all know that the primary function of an axon is conduction of action potentials. That's what we know, and that's how the entire nervous system is built upon. These neural networks, beautiful neural networks, which fire action potentials, and that's the reason why we exist, why we think, and all these other different neural mechanisms that takes place. But in addition to conduction of action potentials, within the axon, there are cellular mechanisms in place that move. Normally, they move cellular organelles. For example, mitochondria. We have these wonderful dynein motors, which go on these proteins called as microtubules. They look like railroad tracks, where these motors move mitochondria and other organelles of the cell all along the axon. So that's a very interesting observation, because that is what we want to see if we can make use of, if at all we want to transport therapeutic cargoes. And this mechanism is fairly robust, and it's conserved through species. The mouse, the rats, all these mammals, they all have this mechanism where these molecular motors within move substances. Normally, they move cellular organelles through these axons. Now, having said that, now we took some motivation from nature. When we thought of nanoparticles moving things into the axon, the first thing the thing that would come to your mind is the rabies virus, if you think about it. Now, you would all be very familiar with the rabies virus pathogenesis. The rabies virus itself is a nanoparticle. So if you look at this illustration here, it's roughly about 180 to 80 nanometer width by length. So it's a nanoparticle, basically. And we all know that when the virus gets transmitted from the saliva of an infected dog, the virus tends to get into the axonal endings. And once it's in the axonal endings, it goes all the way up and then gets into the spinal cord and then goes up and into the brain. And we know all that pathogenesis of the rabies virus. But this is interesting that the virus uses this route to take the nanoparticles all the way up. And in fact, there have been some studies recently where people have looked at very small nanoparticles. This is like quantum dots. We call them quantum dots, and magnetic nanoparticles, very, very tiny, in about roughly 3 nanometers or 2 nanometers, 5, kind of in that range, where they see that if you put those, the video I'm going to show you is a video taken within an axon. So as we can see, they travel up very nicely. It's a retrograde transport going from the periphery to the DRGs, but these are very tiny for imaging purposes. And there have been studies where they have tried to put nanoparticles in the vicinity of axons, as this one study has shown so very nicely. And they do see successfully these are all what is called as hollow silica particles, which have entered the peripheral nerve. And all these blue things that you see are axons, but they don't get in as much. They do get into the periphery, but they don't quite get into the substance of the nerve. So there is a bit of challenge there. Now, so far we have seen that the challenge lies in the point that trying to get the nanoparticles into a nerve. We have identified the axon, but how do we get it into the axon? Okay, so one route that we have thought about is the skin. The skin, as you know, has hundreds and thousands of nerve fibers, especially if you think of your palm, which is very, very sensitive. There's a whole bunch of nerve fibers within. So we thought if we could use these nerve fibers and see if we can have the nanoparticles go through them. So the hypothesis was that we'll make these nanoparticles, apply it topically on the skin, and then we could see these nanoparticles go up. And this could have implications in various conditions such as neuropathic pain, neuritis, small fiber diseases, and of such. To test our proof of concept, we have used gold nanoparticles. Obviously, it's not unlikely to put drugs onto gold nanoparticles, but this was a proof of concept study where we wanted to see if the gold nanoparticles would indeed traverse the axons. So before we start this, one of the important things we needed to know was, if we're going to apply gold nanoparticles on the skin, would they even cross the layers of the skin? Okay, we know that the axonal terminals are found within the layers of the skin, but if the nanoparticles cannot traverse the layers of the skin, then this would be useless. So we had to do certain preliminary experiments to see if we can get our nanoparticles into these areas, which is the lower layers of the epidermis and even the dermis, because that's where most of the nerve fibers are. Now, when you look at the section of the skin, this is from the hind paw of a rat. The large pink thing that you're seeing is stratum corneum, the first layer of dead keratinized cells. And that looks like a very intimidating layer if me and you were nanoparticles, that we had to cross this layer and get into the layers of the epidermis and the dermis. So that would be fairly difficult to do. And that is the challenge that lies ahead of nanoparticles. And once you see there, then this is another section we have performed where we have stained the epidermal nerve fibers within. And you see these green wavy structures that you see are embedded within this lower part of the epidermal area. And this is where we are hoping that the the nanoparticles would reach and they would get taken up at this location. So to do that, we synthesized nanoparticles of various different sizes. So in this diagram here, these are all gold nanoparticles of about 20, 105, and 186 nanometer in size. Remember, the rabies virus was roughly around 186 nanometer in length. So we are kind of in the ballpark, and we want to see what these different nanoparticles would do. So before we get into the skin, we wanted to see if they were safe enough. So this is a simple experiment where we expose some skin cells, fibroblast cells, to nanoparticles, and the concentration that we tested seemed to be fairly benign, and we did not see any loss or death of these fibroblasts. So we went ahead and said, like, this is probably they're compatible with the cells, and let's go ahead and see how they penetrate across the skin. So we did this simple experiment where we placed this chamber with the skin across these two chambers that you see. We put the nanoparticles in the upper chamber, and then we see if the nanoparticles come down in the lower chamber after about 3 hours. So this would give us an idea of penetration of nanoparticles across the skin. And for this, of course, we are going to use a post-mortem skin, which has its own weaknesses and disadvantages, but this would be a nice starting place. So what we see is that after 3 hours, in the bottom layer, we start seeing nanoparticles. So on the x-axis, you have the nanoparticle of various sizes that we had synthesized. And on the y-axis is the total number of nanoparticles that we see in the receptor. So there seems to be a size-dependent effect where the smaller size nanoparticles tend to come in a little quicker, actually, and there was a whole lot of them in the lower chamber as compared to the others. But the take-home point was, when we took this post-mortem skin and applied nanoparticles on the top, they seemed to go through the layers of the skin, and this was very encouraging. So we went ahead and did this in a live animal. So we anesthetized a rat and exposed its hind feet to solution of gold nanoparticles. This pinkish solution that you see is actually solution of gold nanoparticles. And what we did see is that after about 3 hours, I'll draw your attention to this particular figure here, panel C, where we are trying to look at the concentration, the number of nanoparticles in blood after 3 hours of exposure to the exposure in the animal and after 4 days. Okay. So the nanoparticles were exposed for only 3 hours. And after 3 hours, we checked the blood for nanoparticles. We let the animal be. And 4 days later, we came back and then we tested its blood again. And then we saw that it took 4 days for the nanoparticles. On the 4th day is when the nanoparticles started appearing in the blood. And what's also interesting is that we started seeing accumulation of nanoparticles in the skin itself. Within the skin, we started seeing accumulation of nanoparticles. All this was fairly good because this tells us that there is a small component of nanoparticles that stay in the skin, and it appears that the skin acts like a depot, and then the nanoparticles come into the blood. So at least they are traversing the skin. So when we look at the skin, these are sections of skin taken from those rats that I showed you previously. After 3 hours and 4 hours after 4 days after nanoparticle application. So we use this technique called as silver augmentation. We amplify the gold nanoparticle signals because the gold nanoparticles are so tiny that you cannot visualize them in regular microscopy. So we have to amplify the signal using certain silver dyes. And what you see here, these black components that you see, are actually representative of gold nanoparticles sitting in those areas. And we were fairly encouraged because if you look at these pictures here, especially on the bottom, the dark brown is the stratum corneum, and you have all these black sediments here that's sitting in the nanoparticle, in the epidermal and lower epidermal area. So this basically says that they're all in the vicinity of the axons, because the nerve terminals that we discussed before are generally in the epidermal and dermal areas. So this was very interesting to us. So we want to go further, and then we wanted to try these nanoparticles in as a topical application and see how this goes. To do that, we first modified these nanoparticles a little bit. So we modified in the way that we put these moieties, what is called as isolectin B4. This is a very interesting molecule we find in plants. And this tends to bind only to unmyelinated nerve fibers. So C fibers. It tends to bind pain fibers and unmyelinated fibers. So we put this protein all around the nanoparticle. You can see in this image here, there is a very thin coating of IB4 that has come up on these nanoparticles. The reason why we do this is we are trying to make this nanoparticle bind to neurons. So since isolectin B4 binds to neuronal membrane, we have put in this on the particles. And then we isolated some DRG neurons and then dorsal root ganglion neurons. And we just wanted to test if this nanoparticle binds to these neurons. So what you see in these images are just cell bodies of DRG neurons. And we see that these particles very nicely goes and binds to these neurons. The red indicates the light scatter coming from the nanoparticle, and the green signal is coming from the isolectin B4 molecule itself. So when you see yellow color, it means that it's a merged image, which says that, look, the IB4 containing gold nanoparticles have all entered the neuron. And what is also interesting is if you see this picture, there are two neurons here. There is one which has the yellowish greenish stain, and the other one has nothing. So this also kind of tells us that the nanoparticles that we have synthesized is very specific for the type of neurons that bind to isolectin B4. And as I mentioned, these neurons are the ones that bind it's this unmyelinated C fibers. The neurons that give out the C fibers are predominantly the ones that bind to isolectin B4. Don't ask me why, but that's how it's in nature. If for some reason these neurons, the unmyelinated neurons, they bind to the isolectin B4. So these experiments suggested that the gold nanoparticles that we made did bind very specifically to these DRG neurons. But the point we are using them is at the cell body level and not the axon yet. Okay. Now, when we did a simple electron microscopy image, we saw these nanoparticles nicely. We can even see them entering, and they're forming an endocyte within, and then entering via endocytosis. And you see that these nanoparticles are inside the neuronal cell body, confirming that the nanoparticles do enter neurons. Now we wanted to test these on axons, because the previous experiments, we just wanted to try if the synthesized nanoparticles bind to neuronal cell bodies, and neuronal cell bodies are fairly easy to culture and do all that. But now the hard part comes in, where we wanted to test it on axons. So for that, one of the challenges that we faced was to spatially separate the cell body and the axon, so that we can apply nanoparticles on the axonal side and see if they get taken up and if they go into the cell body. That's the challenge we wanted to do. So we used a microfluidic system. What you see here is a microfluidic system where you culture axonal cell bodies on one side, and after about 3 or 4 days, the axons come out from the cell bodies, just like the way we saw in the video that I had showed, and they go through these micro grooves that you see here, and they get on the other side of the compartment. So this gives us a beautiful system where we can apply nanoparticles in one chamber, like here, and then we can observe the nanoparticles go through the axons and into the cell body, if at all they do it. And here are a few representative images of DRG neuron cell body, which are sitting on in the cell body compartment. And you can see these nice axons that have come through the grooves and have come through the other compartment. So now this gives us an opportunity to test our nanoparticles in this chamber here. So that's what we did. We applied these isolectin B4 conjugated gold nanoparticles into the axonal compartment, and then we put these devices under confocal microscope, and then we started visualizing these axons. The green image is just a marker for an axon. So wherever you see green, it means the axon is present over there. But the red signals are coming from the gold nanoparticles. So as you can see, these are images taken from the same axon. And what you see in the third panel, C panel here, is that I would draw your attention to these spots of yellow. So the yellow basically means it's merging, the red signals are merging with the green signals. And that's where you get these yellow signals. And these happen to be all along the axon. And so this got us all very excited, because this basically means that the nanoparticles are associated with the axon as they go through. But however, with this data, it's hard to say if they are inside the axon, or they're just sticking on top of the axon. So for that, we do a bit of what is called as optical sectioning within these images, and we kind of see these yellow colors inside the green. You know, if you observe these, there's yellow color inside the green. So this kind of suggests that the nanoparticle may be within the axon. And these images here are just control images where we have used nanoparticles, gold nanoparticles, without putting these isolectin B4 on top of them. And when you just put plain nanoparticles, they do not seem to be taken up by axons. It's only when we put the IB4, isolectin B4 on the nanoparticles, do they get taken up by the axons. Further, we did some quantification of these data. So what we are trying to see here, we have used this software which tries to quantify these correlations where we have merged these images. It tries to give a number to it. And so what you see here again, as before, is these spots that you see, the hot spots that you see, basically means that there are the signals have merged with the gold nanoparticle signals have merged with the axon signals. And we get a nice correlation index of 0.51. But when you look at the controls of only gold nanoparticles without isolectin B4, the correlation index is fairly low. So this suggests that the gold nanoparticles, isolectin B4 conjugated gold nanoparticles, do indeed go into the axon. But this is not enough. We had to further confirm this finding. And so one of the way we did it was we used the same microfluidic device, and then we started imaging the cell bodies continuously, live cell imaging, basically over a period of 6 hours. What you see on this graph here on the x-axis is time in 6 hours, and on the y-axis is the change in fluorescence. So what does this mean? So if you put nanoparticles on the axonal compartment, they will slowly go in and go into the cell bodies over a period of time. As they come into the cell body, the fluorescence will start increasing within it, because the more the number of particles comes into the cell body, the more the fluorescence. And that is what is being graphed here. I'll draw your attention to the circle, the dark black circle here. Over a period of time, when we added the IB4 AUNPs, so we added the gold nanoparticles with the isolectin B4, the fluorescence intensity gradually starts going up in the cell body, which suggests that the nanoparticles are moving through the axon and coming into the cell body. However, when we added a drug called colchicine that messes up the microtubule within the axon, we did not see this increase in fluorescence. So the triangles are the ones where we have applied the colchicine. So now this is again a confirmatory data saying that the nanoparticles that we are putting at the tip of the axon are indeed going via the axon, probably using the microtubular system and getting into the cell body. So we even scraped out the cells from the cell body side and performed a transmission electron microscopy. And we see that the nanoparticles are indeed inside the cell bodies of those neurons. And this was excellent and very strong evidence for us to go further and state that the nanoparticles that we have prepared does go inside the axons and reach the cell body. But the experiment that we have done is very, very, it's in an in vitro system. The distance is only 150 microns. The micro grooves that we're talking about is only 150 microns. So now we have to test it in a live animal. So for that, we first wanted to confirm if these nanoparticles will bind the intraepidermal nerve fibers, which are present within the skin. So we applied these nanoparticles on isolated skin sections, and lo and behold, we see that these signals, what you see here, are nanoparticles binding to intraepidermal nerve fibers within the skin. And this was also very encouraging because now we had evidence that the nanoparticles can traverse through the axons and they can also bind epidermal nerve fibers. So we finally wanted to come up and show it in an animal model if this is indeed the case. So we exposed the hindfoot of a rat, live rat, to our nanoparticles. These are the isolectin B4 conjugated nanoparticles. And then we wanted to see if the nanoparticles will they come up in the DRT? Because this is the final test, actually showing it in a biological system. And what we did see, I'll draw your attention to this graph here first, panel C. What we did see is that if we put nanoparticles without the isolectin B4 conjugation, we have a baseline level of nanoparticles that come up that come up in the DRG, in the sciatic nerve, and on both the ipsilateral and the contralateral side. This could be probably because many of these nanoparticles will definitely enter into the bloodstream once we apply into the skin. So that is probably what we are seeing here. But what was very interesting was that when we look at the DRGs from the ipsilateral side, on the same side as we put the gold solution, gold nanoparticle solution, we see that there is a significant increase, a considerable increase in the presence of nanoparticles on the ipsilateral side of the DRG. And this was very, very exciting for us because now we have shown that the nanoparticles do enter via the skin, presumably via the sciatic nerve, and they enter the DRG. However, we did not see, or we did not do experiments to see if they do go ahead and go into the dorsal horn. That is something that we want to do in the future. Now, what is also important is that interestingly, what we saw is that if you add the isolectin B4 to the gold nanoparticles, the number of nanoparticles that actually cross the skin is reduced. So this in one way is good because you would want to reduce the toxicity of nanoparticles when you apply into the skin. You want them as less as possible to get into the blood, because once they get into the blood, as all of you know, it'll go everywhere, liver, kidney, and stuff. They're benign, but still, you wouldn't want them floating around in your body. So that was one very interesting finding that we saw, that if you conjugate them, they cross less across the skin. But whatever the IB4 AUNPs cross, a significant number of them do get into the DRG. And so finally, of course, putting gold nanoparticles inside axons, even though it might look fairly impressive to us, we have to then look at the function. Does this hinder nerve function once we put it in? So we did a simple electromyograph where the recording electrode was placed here and the stimulating electrodes are placed in these areas. And we did not find any changes in nerve conduction studies. So all these waves that you see basically says that, look, there's no difference between the controls. So this suggested that what we saw at the concentration that we have used, the nanoparticles in the concentration that we have used, does not seem to affect nerve function. So to summarize, I hope I have time. Okay, I think I'm done. This is the last one. To summarize, we conjugated IB4, we synthesized IB4 conjugated gold nanoparticles, and we showed that they could bind to the neuronal membrane. Then we saw that they could even be internalized by axons, and they undergo retrograde axonal transport into the neuronal cell soma. And importantly, topically applied gold nanoparticles can bind to intraepidermal nerve fibers, and they can also translocate all the way through the nerve fibers and end up in the ipsilateral dorsal root ganglia. And we are hoping that in the next experiments that we are doing, we are trying to make particles where we can actually put drugs into them for some of these diseases, such as neuropathic pain, neuritis, small fiber diseases. So it has a lot of applications there. In addition to it, we can think of applications in neurosurgical cases where axons are accessible, and if that can be sent in to their cell bodies, maybe for diseases, Parkinson's or any other kind of diseases. We haven't looked into those kind of things yet. We're still in the peripheral nervous system, and we're trying to see if these play a role. So with that, I would like to end my talk. I would thank you all for your attention. And if there's any questions, please go ahead and you can ask. Thank you very much.
Thank you, Dr. Sahadev. That was a very interesting talk. And the title you chose, Hitchhiker's Shortcut, Hitchhiker's Guide. That's a very interesting title. And we have, and what you have done is phenomena and what you have achieved. So I would like to ask you a few questions. As you know that I am not a medical person and I'm a statistician. My background is statistics. I have done epidemiology, so I understand something. So my questions are going to be very lay, as a lay person asking you questions. So, and I'm sure those in the audience will have more specific questions to your talk. So my question is, one is that why did you choose gold as your nanoparticle?
Yeah. Thanks for that question. One of the easiest answer is that they are easy to synthesize and they are easy to manipulate in terms of size. Whereas the other ones, because ideally, if you would say from a application point of view, we should be doing this in certain polymers. So there's something called PLGA polymers, which are FDA approved and can be done. But manipulating them and getting their sizes is fairly difficult. So gold gives us this advantage that it's easier to get their size. Now, once we have a proof of concept and once we have convinced ourselves that this route does indeed exist, now the next thing is to go into those kind of polymers where you can actually put in these drugs and send them across. But that's one of the reasons for using gold.
The reason I asked that was because in Ayurveda they used heavy metals, and we have been the greatest accusation against Ayurvedic medicine is heavy metals, and here you're using gold, and so would that become an issue, or do you think there is some truth in what the Ayurvedic people are doing? Are they able to achieve what? It's not that we have any experiments for it, but do you think there's some justification in what they're doing and using this kind of topical medicines, and when they claim that it will reach all these neuropathies they're able to heal, it seems now that they're justified in what they're saying, just because you have now experimentally shown it, what they have been doing for hundreds of years. What is your comment on that?
Yeah, so very, very, very valid question on this. So as you rightly pointed out, gold colloids are used in Ayurveda for various reasons. I'm not an expert on that, but what you say is correct, that they are using it. Now, in this study, one of the interesting points was that the gold that we used in the concentration that we have used, the nanoparticles in the concentration that we have used, was not toxic. So that was one of the things. I mean, the data cannot vouch for any therapeutic or enhanced benefit. That we do not have the data for. But at least in the concentration we have used, we see that it is not toxic, especially even when it goes into the axon. So you could imagine that it's going into the cell body of a neuron. There would be some kind of a sensory deficit, which we did not see. Even when we observe the rats, usually they have certain behavioral phenotypes that can come up when there are sensory deficits or even pain. There are certain pain behaviors that come up. And we did not see that in these animals. So at least what I can comment is about the safety in the concentration that we have used. Maybe higher concentration, probably it might cause, I mean, of course, concentration, whenever you play with concentration, higher concentration always has certain effects. But at least gold in this concentration did not, even when it got into a neuron. So I would stop at that particular comment, because we haven't done any experiments to say the benefit of it. But at least it was benign that way,
And the other thing is that You know how blood lead is such a big issue, and we have, at least in the US, they have done everything to stop lead from the atmosphere. So they've taken it out of, gas or petrol, and out of, uh, plumbing and everything, so that the lead is not absorbed into the body. And lead, again, that's again a nanoparticle, and it's also another metal. And there's so much. And of course, you did mention now that the concentration at which you have done, there is no side effects. And but lead has been again another um, metal that is highly used in the Indian system, and has been condemned. So apart from gold, what other metal would you consider? Um, I mean, I know that Ayurveda uses lead. What other? Um, is only gold the option? Is my question actually.
Yeah, from a nanoparticle perspective, so from what we have been doing so far, the reason why we use gold mostly is for the reasons that I've already mentioned. But there are other metallic nanoparticles that are used. Silver nanoparticle is one that is used. There is platinum that's used. But of course, they have their, we can get into iron oxides and copper, but they all have their toxicity levels, and those are well known. But platinum, titanium, they all show some kind of benign effect to certain levels. But mostly when we talk of especially gold, when it comes to imaging, there's a lot of work that has been done using gold nanoparticles for imaging systems. Like even in this study, when we use gold, we don't use any exogenous fluorescence. It's the light scatter from the gold itself that we measure. So it has these advantages. But after gold, but you raise a very good point about lead, though. I wanted to just comment on that. One of the things that we saw, which was very, standing out in our study, was the way the nanoparticles cross the skin. It appears very easily that they cross the skin and get into blood. So all our fears on when it comes to lead nanoparticles and their concentration, I think, are not misplaced. They travel quite across the skin as well, and especially when we consider children and baby skin, where the cellular contacts are not as tight, you can imagine how these nanoparticles would go through. But yes, that's a valid concern that you've raised. But with the metallic things, I mean, these are the lists as I mentioned, that are being looked at by people. But for most of these kind of purposes, with cellular toxicity studies, or where you want to try these proof of concept, gold is a fairly good option, and fairly easy to do as well. So that's what I would say.
Thank you. I have one more question.
Yeah.
See, we don't have treatment for multiple sclerosis, for example, Parkinson's, and those. Do you hope that we will have such treatments through this method?
Yeah, I would too. I mean, you might be aware that there are these right now, for many of the Parkinson's patients, they're going into DBS, deep brain stimulation, and those are fairly invasive. And one of the things I can think of is if you can find axons, for example, which are fairly accessible. Maybe we can use these less invasive, I wouldn't say non-invasive, I would say less invasive procedures. But at the same time, this has its limitations too, because we do not know what happens once the particles go through an axon and come to a synaptic end. What do they do later? Many theories would suggest that the nanoparticles may get extruded out of an axon at the synapse, and they might get taken up. But that has not been tested. If that would happen, this would stand a good chance where we can send it via axonal tracts in the spinal cord, at least for areas where we want to go. I do not specifically know about multiple sclerosis, because we would have to hit the caudate or specific neurons in specific areas of the brain. But definitely, as a route to enter into the nervous system is something we can definitely study and see if this one would work.
That's interesting. And my last question, and I'm hoping that there'll be questions more from the audience. You were talking about neuropathic pain, right? What about arthritis and pain of those kinds of pain? Would that also be an area that you would consider?
Yes, they would. Arthritis pain, from my understanding, has components of both inflammatory and neuropathic in nature. So for arthritic pains, anti-inflammatory drugs work well. For neuropathic pain, for many patients, the anti-inflammatory drugs don't have that much of a role. So that's why they go into gabapentin and other kind of drugs. And as you saw, that these nanoparticles are going inside a neuron. So many of the receptors for anti-inflammatory drugs tend to be on the surface of the neuron. So one of the drug targets could be corticosteroid receptors. Corticosteroid receptors tend to be within the cytoplasm. So those could be one way of doing it. So as I said, arthritis, I'm not completely sure that arthritis using this approach would be something doable, mainly because of the drug targets that are available that are quite out of the reach of these nanoparticles. But yeah.
Okay, thank you very much. And I think we have one comment here, one or two, and we hope there will be more here. So here's a question from Varsha Srinivasan, and she was asking, how will the nanoparticles defy the immune response of the host? Should the host be put on a course of steroids?
Oh, what a wonderful question. So in our studies that we have seen, these were short duration studies where the axons, where the gold nanoparticles are inside an axon. So when they're inside, they do not come in contact with immune cells. However, we do see that prolonged periods of stay of nanoparticles within tissues do have elicit an immune response, but we haven't seen it to an extent where they would actually need a course of immunomodulation. So that we haven't seen. So my guess is, I'm using the word guess because we haven't done long-term studies to actually follow and look at immune cells and their characterization. It did not appear to have any of those systemic effects where immune response would have been exaggerated. So at this point, it seems fairly benign that we put in, and it does not evoke any immune response. But in cellular studies, we see that the macrophages take it up. When we just do it in vivo, macrophages do take it up. I'm not talking about the axonal study, but I'm just saying generally that the macrophages do take it up, and then they're extruded out, and we don't see a whole bunch of robust immune response. We don't see that with metallic gold nanoparticles. Yeah.
Yeah. That's very hopeful that when, and we have one more question also from Vasha. Why choose only DRGs? Why not other sympathetic and parasympathetic ganglia?
Yeah, we would have loved to, but the access to those ganglia, especially when we're doing it in a rat and a mouse, is fairly hard. And the innervation from those is a little more difficult as compared to a nice DRG when you take it when you look at the innervation into the hind paw, and then they nicely go into the L3, L4 area, and you can dissect those DRGs. So that gives a very nice system to study this process. But after that, I don't see why it can't be tested in any other axons like sympathetic and parasympathetic. So to answer this question in short, it's primarily because of the ease and the beautiful innervation that we see with a DRG. That's the primary reason why we chose to do this proof of concept study in a DRG neuron.
Thank you.
Okay.
If anybody else has any question, please post it. on the chat, and we will be able to answer you.
There is one question by Dr. Cletus. Ma'am, did we address it? I think we did.
No,
right?
I didn't see it. I'm not able to see Dr. Cletus's question.
All right. So what is the possibility of immune reactions against nanoparticles by the host, especially against conjugated NP? This is the question by one of our researchers and assistant professors of biochemistry.
Right. As I mentioned, at least in what we have seen with metallic nanoparticles and even putting in the IB4, IB4 is by moiety by itself has been in use for a long time. People have even injected IB4 for various staining purposes and things. And there doesn't seem to be any reports of immune reaction coming up that I know of for that particular molecule at high concentration. But in the nanoparticle domain, we have been using many of those polymeric nanoparticles, platinum nanoparticles, gold, and the immune response is not robust. I mean, I will not say it does not exist. Obviously, anything that we put into the body, there will be a mild immune response. And we have seen that. I think there are some FACS reports, FACS data, flow cytometry data, where we see certain elevation of T cells, if I remember right, when we give these nanoparticles. But that hasn't deterred us from getting into these therapies because they don't seem to play that big of a hindrance yet. Of course, if you modulate certain chemical moieties on nanoparticles, they do. But generally, we have used certain PEG molecules on polymeric nanoparticles, and those seem to do well. And we don't see a whole lot of immune response from them. But if we want to, for experimental purposes, get immune responses, then that is easily possible with nanoparticles. When you put certain molecules on top, you get them. But generally, when we are trying to do something for a clinical application, there are strategies available where we can decrease the immune response. And in this experiment, thankfully, even when we put the IB4 and we let the animal go for 4 to 5 days, we did not see any response, because even if it's usually an acute response, we should have seen something by then. And we did not see that. Even the nerve was healthy, so we didn't see any response.
Thank you, sir. One question from one of our students, factoring in the capacity of nanoparticles to utilize the mobility in axons, do you envisage this particular modality as a potential route for drug delivery?
Yes, that's where we are heading to, actually. I have one of our students who's trying it in, I had mentioned polymeric nanoparticles, where we can actually put drugs inside the particle. And we are hoping that we can even show if we can put drugs into the neurons. Once we show that, then the applications will open up for a specific disease, a specific condition, and things. So that is the idea, and we want to go there.
Excellent, sir. And one more question. Can this technique be used for diagnostic purposes also?
Yes, absolutely. In fact, there is, I think I spoke to one of my colleagues in a company where they are trying to see if they can use certain nanoparticles for visualizing the innervation into lumbar DRG, especially when it comes to backaches and trauma, crushing trauma that happens at those levels. So they are looking into diagnostic, especially imaging purposes, to see how these things are. Yes, definitely.
Have you explored any scope for adverse reactions due to these initiatives? Has it shown any signs of adverse reactions in the animals?
Right. So we have only looked at two aspects of the adverse reaction that you say. One was the behavioral, because there are certain traits which we can pick up when we look at an animal, if it's in pain or other things, which we did not. But the second one was what we thought was, if it was to have an adverse effect, especially in a neuron, the first thing that would affect is the conduction, the nerve conduction. And that's why we did this a nerve conduction study. It was an EMG study that we did. And we did not see any change in the conduction velocity once we did those studies, the electrophysiology studies. So, which is where we concluded that the concentration, I keep harping on the concentration, because we fear if we increase the concentration, it might affect the axon. Because we are putting in these particles, the metallic particles in. But at least in the concentration that we have utilized, it did not seem to change the nerve conduction. But now going into therapeutic application, if we do produce polymeric nanoparticles and we try it, even then, we have to be careful with the concentration. Anything higher, that might have a detrimental effect on the way nerve functions.
Thank you so much. So that's quite encouraging from that perspective. And before we end, this is a special request from one of our senior students. So he asked me personally, so will you please ask him how he made his way to Harvard? He's more interested in that. So I guess he's there. He's one among the audience. So he was very particular about that. And just to add on to that, how from MBBS you moved on to research, and what was your inspiration, and how can, as young students, they too aspire the way you did, and what are the avenues? Can you just give us a glimpse of your life journey, sir?
Yeah, I could do that. But if you're expecting inspirational, all these, I'll tell you the truth. So when I was doing my MBBS, I was a very slow clinical practitioner. So by the time the symptoms, I pick up the symptoms and the sign, and by the time these disease differential diagnosis comes in, the patient would be bored and would want to go. So I was fairly slow in doing that. And what the beauty of research is that it gives you this time to sit and think about stuff. And that is what was exciting for me. Especially, I mean, each one of us have our own personalities and our strengths and weaknesses. And one of the things that I would love to do is when you get a sign and symptom, I would definitely want to open my physiology book and see why that is happening. But when you're a clinician at a table, you don't have that thing to sit and think about that receptor, those things are not there. So I would say from the beginning, it was very fascinating to look at the mechanism and the pathogenesis of the disease. And having done MBBS in India back there, obviously in the 90s, not much focus was given on research. And I must confess, I didn't even read a single research paper when I was in MBBS, because it was all very towards clinical practice and all that. This was in the 90s. Once I went to the United States, because at that time when I went to the US, I wanted to get into public health, community medicine kind of a thing. And I ran out of money. There was no money, So then one of the jobs that I took up was as a lab tech. I took up a job as a lab tech. And that was my first exposure to research, hands-on bench research. And I just loved it. We were doing these it was molecular epidemiology. Dr. Alice might be interested. So it was a molecular epidemiology where we were looking at these genes and stuff. And that was fascinating. I think that was it. When I once went in there, at that time, I wanted to do my USMLE, and I even did my USMLE. I cleared all those. But then I got caught in the research web. It was so amazing that I went on to do my PhD in neuroscience. And then, I wasn't even actually focusing on Harvard and MIT. It was just that there were some very good investigators who were doing these work, and I was fascinated by what they were doing. And I just wrote to them, and I was lucky enough that they thought it was good enough that they could pick up and I could come work in their lab. So it's as simple as that. I was just doing what I like to do, and things just happened that way. That's all I can say. I mean, there's nothing fancy about it,
Thank you, sir. Thank you for being so candid and so humble. Uh, well, destiny is also a very important
Oh, absolutely.
factor in
Absolutely.
shaping our future, isn't it?
Absolutely.
So, but I'm very sure that you are being very humble. Anyways,
I would just add that if you're determined, you can do it.
Oh, true. When we run out of money, those things come out automatically.
thank you so much, sir. Thank you. That was really an eye opener of a talk, and I think our students and other participants are really blessed to have this talk from you. And I think it's been in the bucket for almost 2-3 years ever since we met,
Yes, yes.
isn't it, sir? And I was instantly impressed by the way you showed around your lab and how you have set up the whole system. And it's really a wonderful work that you're doing at Amrita. And all the best for all the research that you do, sir. And looking forward to coming and meeting you also in the future. And I would encourage our students interested in research to get in touch with you. I hope you would love to have them. And our students have these peripheral postings. So especially with the CBME curriculum coming in, we have these peripheral postings, and I would love to link up a few students with you if you would like that, sir.
Absolutely. Please share my email address with your students if they have any questions or anything. I would love to help them out, address whatever they have. I think you have my email address.
Yeah, I have that.
So so do share. I would be very happy. And thank you for having me over. This was very. I enjoyed it. I enjoyed it. Thank you, Dr. Alice. Your questions, some of those beautiful questions. I loved addressing them. Thank you.
Thank you. Thank you for coming in.
Thank you so much.
All
thank
right,
you, sir.
all
And thank you, Alice, ma'am, for moderating this session quite admirably, and all our participants at the webinar. And just to mention this, we have collaborated with the Students Network Organization, an international student body, and this has happened just yesterday. And I really hope that all the webinars in the future would be in collaboration with the SNO, and it's a good news for the whole learning process. So with that, let me sign off from Believers Church Medical College and Hospital. On the 16th, we have an amazing webinar on CRISPR. You would have heard about the gene process, the genetic modification process called CRISPR. You would have seen it in movies. So it's going to be a webinar on CRISPR. It's at 8 a.m. I'm not sure how many of you would manage. We are looking at a different viewership apart from our students. So whoever is, it's possible, whoever is having a free time, please do log in. It's on the 16th at 8 a.m. and looking forward to have you all for that webinar as well. And until then, let us sign off from Believers Church Medical College and Hospital. Thank you so much. Thank you.
Bye.